Hello again, this is video number 8 for the immune system, and in this video I'm going to discuss the activation and effector response of the cytotoxic T-cells. Previous two videos were the other lymphocytes, B cells, and helper T-cells, and this one will discuss cytotoxic T-cells. So just to remind you, we are talking about these 4 steps. What causes activation? What happens as a result, clonal expansion, and what is the effector response for a cytotoxic T cell? And also for cytotoxic T cells, like the other two we've talked about already, B cells and helper T cells, memory cells are generated. Okay? And this slide is just to remind you what a immunocompetent cytotoxic T cell looks like. So it expresses a receptor with a co-receptor. The receptor is an antigen receptor, T cell receptor here, unique for every immunocompetent T cell. And it's coupled with a co-receptor for cytotoxic T cells. It's the CD8 protein co-receptor. Okay? This combination of receptor and co-receptor can bind to cells that have a foreign antigen matching the antigen receptor, and also carry that antigen coupled to MHC1. And that's because CD8 recognizes and binds to MHC1. So that combination, foreign antigen coupled to MHC1, will be found on body cells. Any nucleated body cell is going to have that combination, because all body cells with a nucleus constantly do this. They take internal structures as a normal process to break some down. Even if the cell is not dying, right, they're processing normally, breaking down components and expressing them on their surface. So they express them on their surface coupled to MHC1 antigen. All right, so I want to talk about the activation process for cytotoxic T lymphocytes, and I made a short video to describe that. So let's look at that first, and then we'll come back to the PowerPoint and review what's in that video. All right, let's talk about how cytotoxic T cells are activated. Cytotoxic T cells have a T cell receptor, and they have a CD8 co-receptor. So on the surface of a cytotoxic T cells, you have both of those. And here I've indicated the T cell receptor as 2 blue lines and the CD8 co-receptor as this red line. Okay, so receptor and co-receptor, and that's what's making up the, this would be CD8, right? And this would be the T cell receptor. Cytotoxic T cells have that combination, T cell receptor with CD8 co-receptor. Now, if a cytotoxic T cell is in a lymphatic organ and it experiences a body cell, any kind of body cell, remember body cells have MHC1, and they couple internal antigens to MHC1 and put it on the surface of the cell. And that's how immune system cells can tell what's inside this cell. Now, if this is, let's say this is a normal cell. It has only normal self-antigens inside of it. And so it's going to put on the surface only normal self-antigens. Okay, there shouldn't be a cytotoxic T cell that would recognize normal self-antigens, because those got removed during maturation, during the negative selection process, okay? However, let's say you have a body cell that is abnormal. And in this case, let's say it's virus infected. Here's a virus, gets into this cell, and then it's going to start taking over the machinery, producing viral particles, viral components. So this cell is going to do the same thing as any body cell does. It's going to take pieces of what's inside of it, stick it on the surface along with MHC1. So it has a lot of normal self-antigens on its surface, but it also is going to be showing some viral antigen. This is not going to kill the viruses. They're going to be replicating really rapidly, but some of them will get broken down and coupled with MHC1 and put on the surface. Well, if along comes a cytotoxic T-cell, it can recognize this combination. Cytotoxic T-cells, the TCR is an antigen receptor. If that matches with the foreign antigen, that's step one. Then the co-receptor CD8 matches with MHC1. That's step two. If both of those match, then that's going to activate this cytotoxic T-cell. If there's not a match of both of them, one of them could match, okay, but not both of them, then there will be no activation. However, if there's a match, this T-cell now has been selected for activation, and it's going to be called into play to do battle with this abnormal cell and maybe any other abnormal cell also harboring this virus that's spread a little bit, let's say. So what happens when a cytotoxic T-cell gets activated? First, it has to bind to the foreign antigen, and like the helper T-cell, it's picky how antigens are presented. It can't just be a virus particle or a bacterium. It must be a foreign antigen coupled with MHC1 on the surface of an abnormal body cell. Okay? If it's a match, they both bind, then this T-cell is going to undergo clonal expansion. It's going to divide very rapidly. It's going to form thousands of similar cells that carry the same combination, this specific T-cell receptor and the CD8 co-receptor. From this expanded pool of cells, you'll get mostly effector cells and some memory cells. What do the effector cells do? So the effector cells secrete a combination of two proteins, perforins and granzymes. Perforins are going to punch holes in this cell. They perforate it. And then the granzymes are going to enter, and they're basically enzymes that are going to degrade this cell from the inside. So this is a 1-2 punch. Punch holes in it, and then allow enzymes to enter it and degrade it from the inside. The cell is going to undergo apoptosis, and it's going to die. And then along will come a macrophage, take it in, and degrade it, okay? The memory cells are going to be basically clones of this cell. Same exact T-cell receptor and co-receptor, and they're going to be stored away in your lymphatic organs and tissues in case you meet this virus again somewhere down the road. And then you'll be armed with many more of this cell with the same antigen specificity than you were the first time you saw it. Okay? So that's how a cytotoxic T-cell gets activated, and that's what it does when activated. Okay, activation of cytotoxic T-cells then requires stimulation, first of all, by binding to its specific foreign receptor antigen through the antigen receptor, the TCR, on the surface of the T-cell. Binding is TCR to the antigen and the co-receptor, CD8, to MHC1. So cytotoxic T-cells can only be activated by antigen being presented coupled with MHC1 on the surface of an abnormal cell. There is a second stimulation as well, and I mentioned this a bit in the previous video, and this is by a boost, basically, from helper T cells. They secrete cytokines, which are going to stimulate activated cytotoxic T cells to undergo rapid cell division during clonal expansion. Okay, first stimulus is binding to the foreign antigen. Second is stimulation by helper T cells. All right, after stimulation occurs, then like for both of the other two types of lymphocytes we've talked about, there's clonal expansion. So that particular T cell containing that specific antigen receptor is going to expand to a huge pool of cells now. And they all contain the same identical antigen receptor. Most of them are going to become effector T cells and carry out the immune response against the abnormal cell or other cells like it in the body. Others will not become effectors, but they'll be stored away as memory T cells for maybe a subsequent exposure to this same foreign antigen. All right, so this slide is summarizing what happens in T-cell, cytotoxic T-cell activation step. And here is the cytotoxic T-cell carrying an antigen receptor and CD8. And here is so this should be a normal body cell with MHC1 and some foreign antigen coupled to it. And let's say it's a viral antigen. And here's match, binding of antigen receptor to the foreign antigen and CD8 binding to MHC1. Okay, so that's the first thing. And the other part in the second signal and stimulation is IL-2, a cytokine coming from a helper T cell. Right, when that happens, this undergoes clonal expansion. Here are just 3, but that'll be 3 of thousands of cells resulting by rapid cell division. And of these, most will be the effector cells, some will be the memory cells. What do the effector cells do? So the effector cells are going to leave the site of where they're generated and go to the actual site of infection. So the activation occurs in the lymphatic, secondary lymphatic organ or tissue, let's say the lymph node or the spleen. But after activation, then the effector cells are going to go to migrate through the bloodstream or maybe the lymph to the actual site of infection. And that's where they'll encounter the abnormal cell, most likely. They're going to destroy these abnormal cells. We're talking about virus-infected cells, but another type could be cancerous cells that are displaying this foreign antigen. Okay? So how does that work exactly? The 1-2 punch that I described in the video. When they encounter an abnormal cell, they're going to bind onto it, again, through the T-cell receptor, antigen receptor, and the co-receptor, CD8. And then they're going to release 2 types of proteins, perforins, which perforate the target cell, plasma membrane, and granzymes that start to degrade it. The target cell, the abnormal cell, undergoes apoptosis, and it will be taken up by a macrophage, which is likely also to be present in the cell. By the way, this is true for the T-cell response and also the the cytotoxic and the helper T-cell response. Cells themselves are doing the action, and that's why it's called the cell-mediated immune response. All right, so this slide is summarizing the effector response of a cytotoxic T-cell after it's activated and it bumps into an abnormal cell having the same combination of foreign antigen matching the antigen receptor and MHC1. Then this cell secretes perforins, so they're represented by these spike-like proteins here, which assemble and puncture a hole in the abnormal cell membrane. And granzymes that enter through this perforation and start degrading the cell from the interior. The cell degenerates, undergoes apoptosis, and will eventually be taken up by a macrophage, and which will complete the process. Notice that this is the same mechanism by which natural killer cells kill their target cells, using perforins and granzymes. Natural killer cells are not specific, though. That is, they have a non-specific type of recognition of abnormal cells, whereas cytotoxic T cells specifically recognize through binding of an antigen receptor to a non-specific, to an abnormal foreign antigen, whether this is an abnormal cell or not. So even though the mechanism of action is the same, the way they recognize abnormal cells is slightly different. Specific for cytotoxic T cells, meaning specific antigen recognition. All right, this is the last slide, which kind of summarizes lymphocyte activation and effector response for all of the three cell types we've talked about. The immunocompetent B, helper T, and cytotoxic T-cells reside in secondary lymphatic organs or tissues, such as lymph node, spleen, tonsils, or MALT. That's the site where they'll encounter a foreign antigen, and that's the site where they will be activated and undergo the clonal expansion. After that, they exit from the lymphatic organ, and they travel to the site of the infection, usually attracted by pro-inflammatory chemicals to the site of the infection. So this is where they carry out their effector response. Helper T cells release cytokines stimulating other immune cells. Cytotoxic T cells will release perforin granzymes to kill abnormal cells. And plasma cells secrete antibodies. The plasma cells don't migrate, but the antibodies do. They'll show up at a site of infection, and they will bind to the antigen that they recognize, thus tagging it for destruction. All right, I'm going to end this video here, and there will be one or possibly two more videos kind of summing up the immune system, so I'll be posting those soon as well. See ya.